Hello and welcome to today's webinar, Updated Mandatory Guidelines for Federal Workplace Drug Testing Programs, What and Why. During today's presentation, attendees will be in a listen-only mode. If during the program you would like to submit a question, please use the chat pod, which you'll find located in the lower left-hand corner of your screen. Type your question in the box at the bottom and click Send. If you have any technical difficulties, you may contact the Help Desk, and that number is 877-297-2901. Now I'll turn the program over to Julian Hoffman. Julian, please go ahead.
Hi, thank you. Good afternoon, everybody, and thank you for joining us for our NSC Government Affairs webinar about the updated mandatory guidelines for federal workplace drug testing programs. We're really excited to have so many of you registered and joining us today, and we thank you for taking an hour or so out of your days to join us for this. In the NSC Government Affairs Office, our role is to consistently monitor and engage with legislative and regulatory staff and other stakeholders to move safety forward, and opioids are definitely a part of that. I encourage each and every one of you to sign up for our Government Affairs newsletter at nsc.org/alerts. It's a monthly newsletter. Takes 1 or 2 breaks during the year for summer and winter, depending on goings-on in the federal and state policy worlds. And it includes information on all the most important safety topics that NSC's Government Affairs staff is working on at the federal and state levels. Once again, that's at nsc.org/alerts. Following the presentation today, we're going to have an opportunity for some Q&A with our experts. We'll be taking those online and we'll moderate them for that, and that'll just be dependent on how much time we have at the end. So earlier this year, the U.S. Department of Health and Human Services revised the mandatory guidelines for federal workplace drug testing programs, which was implemented on October 1st. We're having this webinar today because we received several questions from members and others about the decision-making behind this update and what it means for their businesses. And we're happy to have one of the real technical experts behind the scenes, Ron Flegel, with us today. Ron became the Director of the Division of Workplace Programs at the Substance Abuse and Mental Health Services Administration, also known as SAMHSA, in May of 2012. He initially joined the division in September of 2004 as a forensic toxicologist, and before that was with a nationally recognized laboratory. Prior to that experience, he was at a consulting firm working on applied research, technology development, and formulating new proposals and white papers. He has this long background in forensic toxicology, including work at a SAMHSA-certified regulated drug testing laboratory. Through all of this experience and as director of the division, Ron has become a true expert in workplace drug testing policy and played a key role in the development of the mandatory guidelines we're going to be discussing here today. Ron is also joined by Natalie Lembeck, a consultant with the National Laboratory Certification Program at the Research Triangle Institute. As a consultant, Natalie supports multiple projects for SAMHSA, including the drafting of the mandatory guidelines for hair testing. Aside from her role as a consultant for SAMHSA, Natalie works as a researcher at Johns Hopkins University in Baltimore, Maryland, where she currently focuses on the effects of cannabis on the brain and the body. I'd like to thank both Ron and Natalie for agreeing to join us today, and for you all as well. And with that, I'll hand it over to Ron to get started. Ron?
Thank you, Julian. I also want to thank everybody for taking time out of their busy day today to listen to the presentation. I also want to say thank you from the Assistant Secretary for Mental Health and Substance Use, Dr. Eleanor McCants-Katz, who is now as the Assistant Secretary. So with that, I will begin. And as Julian has said, this is basically going to focus on the revisions to the urine mandatory guidelines for federal workplace drug testing, but also it will also look at some of the information that we attained years ago when we started to look at adding the synthetic opioids to the actual federal mandatory guidelines. Under the Federal Drug-Free Workplace Program, the governing authorities are to establish the scientific and technical guidelines for federal workplace drug testing, and also the standards for certification of the laboratories engaged in drug testing for federal agencies. But also in the private sector, there's a number of private sector employers that use the federal guidelines within the testing process, or the laboratories that are actually HHS certified, or private entity laboratories that do the testing on what they call a FUTA basis, which is the forensic urine drug testing analysis. We are governed within the federal program under Public Law 100-71, Executive Order 12564, which was issued by President Reagan at the time. And also we have the mandatory guidelines. Currently, the implementation, as Julian pointed out, was October 1st. The oral fluid are in the proposed final stages, currently hopefully going to Office of Management and Budget here shortly. And also we have a proposed hair drug testing guidelines that is currently being proposed. Under the Drug-Free Workplace program, there's many things that we have to take into account, including federal laws, state laws, testing issues, and also contract and legal issues. And some of these with employer drug testing policies of how they looked at it. And that's really some of the information I want to put forward today so that employers can understand, especially around implementing this synthetic opioid testing as the federal government has done, is what some of the issues you should be aware of. I put this slide on here because within regulation and policy, Office of National Drug Control policy is really the policy arm of what we regulate as far as what we do and regulate under their authority. From the bottom of the pyramid, we also have a drug testing advisory board, which is a scientific and technical board which looks at all the issues within the regulatory programs. We also have an interagency coordinating group executive committee, which is chaired by Office of National Drug Control. And within that committee, we look at a number of issues under the Drug-Free Workplace program, as well as what constitutes a safety-sensitive position, which I think in the private employer are a number of those that are also not necessarily regulated within or outside of the regulations as far as federal or federally regulated, which, just to put that up front, would include federal agencies, which are federally regulated. The federal programs also are, for instance, Department of Transportation and also the Nuclear Regulatory Commission. So the one issue that I looked at when I was setting up this slide is under regulation as policy, is you have donor drug test and the results at the top, but where does the employer follow in regards to that? And I think the employer is really right at the top of the pyramid here because, again, that is a lot around why we do the drug test, whether it's post-accident, reasonable suspicion, it's an applicant test. There's a number of different issues why we do drug tests. So again, I think the employer really falls right on the top of this pyramid. Some of the presentation objectives I just wanted to bring out here. I wanted to look at some of the emerging issues. Again, under Drug-Free Workplace Program, the oversight for the federal executive branch agencies. Some of the studies that we've done around opioid dosing studies and the importance of those studies, why we chose what we chose when it came to the cutoff, quantitative levels, etc. I think that is important, but I also think it's important around policy, but it's also important around how you, or what a medical review officer would say is what is not only a valid test, but what are the results of that test mean. I want to also go over some of the changes to the urine mandatory guidelines, and this is, of course, the inclusion of the semi-synthetic opioids, and also we've added additional specimen validity testing within this implemented mandatory guidelines, and we also removed MDEA from that. Just to speak about that a little bit, is again, within the regulated programs, we saw so few samples that were MDEA positive that we removed that from the screening program, but we did allow the MDA confirmation, which again is a metabolite of MDEA. So we still felt that we would pick up any positive essentially ecstasy samples that we had. And then I want to touch on briefly around the MRO guidance manual and the update. This was really around the review of the semi-synthetic opioid testing in the regulated testing program, specifically for hydrocodone, oxycodone, hydromorphone, and oxymorphone. And again, more of the more common names to those are Dilaudid, Percocet, a number of those that people will hear as common generic names, but again, we focus on the specific analyte. Under emerging issues, really there are several. Within the section of Division of Workplace Programs, we also have part of our office looks at emerging issues, and some of those emerging issues, as you're probably well aware, are the opioids, the marijuana, specifically with all the case or the states moving to legalize marijuana, and also the synthetic drugs. And again, there are a number of synthetic drugs that are moving this direction, and I say that from China, etc., that not only have a potency level much greater than the drugs that we see here, but also they're much cheaper and they're more accessible to people. Essentially, there's synthetic cocaines, amongst a number of those different drugs that we're seeing emerge from China. I also wanted to touch briefly on the fentanyl or heroin use. In the mandatory guidelines that I'll talk about a little bit later, we did not include fentanyl as one of the analytes to test. And I know there's some concern around that because heroin use has increased. But what we do test for in the mandatory guidelines is 6-acetylmorphine, which is a direct marker of heroin use. And so if we do find 6-acetylmorphine, we can go back and look at, on a case-by-case basis, if there was fentanyl in that specimen. So we did a recent study ourselves just to look at in the regulated sector, and we took about 500 samples, regulated samples, ran them through and looked for fentanyl. We actually came up with about 6 positive screens, and 2 of those confirmed for fentanyl. So that was a little surprising to us, because you would think in a regulated sector you would not have fentanyl use. But you do, and that works out, at least in this small sample size, to be, I believe, about 0.6%, which in itself is actually relatively high when you compare it to PCP, which we actually test for in the program. So again, we are looking at this. We're going to do a broader study around fentanyl and look and see if that is maybe one of the emerging issues that we need to look at least in the regulated sector. Also, I included drug overdose as one of the leading causes. In 2015, there was about 52,000 overdose deaths. I believe in 2016, the number I just recently saw is a little over 60,000. So again, significant increase in what we're seeing. Again, just to put this in around all the prescription pain relievers. It is an opioid crisis. Illicit prescription opioid use is now more prevalent in the U.S. than the combined use of heroin, cocaine, and methamphetamine. So when you look at that as a combined, it far outweighs the other drugs or the other analytes that we see. The U.S. is the world's largest consumer of oxycodone per capita. I also had a presentation that I gave. When you look at the number of prescriptions filled, it's staggering, the amount of prescriptions that are filled for oxycodone. The combination product, which is hydrocodone with acetaminophen, is also the most frequently prescribed opioid drug in the United States. Both oxycodone and hydrocodone and other semi-synthetic opioids have become the most commonly misused prescription drug in the U.S. And that's why we focused primarily in the studies that I'm going to show you around oxycodone and hydrocodone. And then again, oxycodone and hydrocodone are now tested in the federal workplace programs and will soon be tested in the federally regulated programs, which are specifically, I mentioned here, as Department of Transportation. They are to implement their guidelines or their DOT program on January 1st, 2018. And again, they're required by law to follow the mandatory guidelines and use the HHS certified laboratories to do that testing. This is just a diagram that shows, I just wanted to point out the top part with the synthetic opioids, and that was other than methadone, is again 20,000 deaths in 2016. But when you combine all the deaths from heroin, synthetic opioids, the natural semi-synthetic opioids, etc., again, I think it's almost 61,000 deaths in 2016 that we saw. So again, I think the concerning part for me here is there doesn't seem to be any leveling off of these lines when it comes to the synthetic opioids. And again, I also want to mention that as we go through the program, if there is a question or a specific question, again, please feel free to put it as a question, and either Natalie or Julian will stop me, and we'll try to answer some of those questions at the time before we get too far into this presentation. I also wanted to mention around alternate matrices. Again, we have a proposed rule for the oral fluid. It's in the final stages for OMB clearance. And there are a number of technical and scientific peer-reviewed journal articles that we've published specifically surrounding oral fluid and specifically focusing on marijuana, because that seems to be one of the issues. The thing around oral fluid is you can see both THC, which is the active ingredient in marijuana which causes impairment, and the THCA, which is the tetrahydrocannabinol carboxylic acid, which is the metabolite in oral fluid. So it's one of the specimens that you can actually see both the parent drug as well as the metabolite. So again, there were some issues around contamination, etc., that I think at this point we've looked at fully in all of our technical and scientific studies, again, for another presentation. But again, that's in the final review process. And again, I think this gives employers another avenue other than urine to test their employees. It's extremely efficient. It's almost as a direct observed collection. We do know in the urine program there are a number of substitution of synthetic products that individuals put into their urine, or they actually just add the synthetic urine itself right to the collection cup, because it is not an observed collection. So again, I think to be a very good program, you need a robust set of different matrices, including oral fluid and possibly hair, to look at to be an effective program. And then, as I mentioned earlier, the hair, we have the first proposed draft and it's been reviewed. We're working with the Drug Testing Advisory Board, subject matter experts, and federal partners on these proposed revisions and recommendations. And then there also is a number of studies that we've started, more to come, around what we're looking at is unique metabolites, specifically around use when it comes to metabolites that are in the hair for that. As mentioned earlier, the mandatory guidelines were published in January 23rd, 2017. The effective date was October 1st. Again, the major changes are listed there. And one of the ones I wanted to focus on here was we also raised the lower pH cutoff levels for adulterated specimen. And again, across the board for all HHS certified laboratories, when that implementation date of October 1st, the laboratories actually changed all parameters on pH as of that date. So whether it was private employer companies or regulated companies, both DOT and HHS or federally regulated samples, that pH changed from 3 to 4. The reason behind the change is, through a number of our studies that we did, we showed that a lot of the synthetic urines that are being substituted in the federal program, the pH levels ran around 3.8, 3.7 to 3.8. So again, in a large population of synthetic samples that we looked at, most of those pHs targeted just above 3, so they didn't meet the earlier criteria within the federally regulated program. So what we did is we looked at a number of normal human urine along with the synthetics, and we were confident with all the clinical testing as well as the federal and federal regulated testing that the pH of 4 was a better target level of how to target actual normal human urine. So we did increase that. So we have seen an uptick in the adulterated samples based on that, but again, a lot of these, as I said, were also synthetic urines that targeted just above 3. DOT's final rule was published in November of 2017, and the implementation date is January 1st. And again, this is just some information that was sent out from DOT, I believe it was last Friday, and it has the DOT's final rule, if you would like to read that, and it also has following links that notice or that clarify the company's policy issues around Part 40, and also within the Federal Drug Testing Custody and Control Form, the use of that new form. And I'll talk about that form briefly because some of the private employers, as they move over to testing for semi-synthetic opioids, we have published on the expiration date for the old form was on May 31st, 2017. We have a new form that includes the semi-synthetic opioids in the chain of custody form. So some of those providers of the forms for the drug testing, meaning the paperless forms or the electronic providers, they will move mostly to electronic forms using the new form probably around January 1st when DOT comes on. Currently, most of the employers, as well as federal and federally regulated, are using the old forms to use these forms up, but also they've been given instructions, sorry, if they do have a positive opioid, how to annotate that positive opioid on the old CCF form. So this is just a little bit of the rundown, the revised urine mandatory guidelines, OMB approval of the expired form. The revised urine mandatory effective date, again, was October 1st. And then DOT's federal CCF expires on January 30, 2018. That is the old form, and at that time, all new forms will replace the old forms. So again, as a private employer, you will start to see those new forms start to show up in the collection sites so that they will begin to use the new forms if the employer are going to the semi-synthetic opioids. I think some of the challenges and opportunities that we've seen in moving all of these forward, including the oral fluid mandatory guidelines, is now there's a little bit more oversight and standardization of the industry to protect donors' rights and validate it with reliable science, and that I think that holds true specifically around oral fluid that, again, is in the final proposed process. The implementation of oral fluid drug testing as an alternate specimen, therefore decreasing the number of substituted and adulterated specimens. Again, it's very difficult to adulterate an oral fluid sample because it's almost as it's a direct observed collection right in front of the collector.
Something that I want to say quickly to address a question that somebody had in the audience as far as the oral fluid sample is concerned. It's not a cotton swab. What we do is actually a tube, and the individual will actually put their spit directly from their mouth in front of the officer into the tube, and that will go off to testing.
Thank you. Thank you, Natalie. Yes, as Natalie said, yeah, and it is a very, what we've said in the standards for that is it's a collection of 1 ml of oral fluid. So it's a very specific collection with an FDA-approved device. And then the last thing would be really one of the hopes in all of this was to deter the illegal drug use or use of drugs and prescription opioids within the federal and federally regulated programs, specifically around safety-sensitive issues or positions. With that, I now want to go into, first I should stop. And Natalie, do you see any other questions before I go on to the actual, some of the studies that we performed?
I think that, yeah, somebody just asked a question regarding the prescription drugs as far as opioids are considered, and why, when they become a violation under the mandatory guidelines and use in the drug, or in the federal workplace.
Okay. Real briefly, after we go, well, I'll answer it real quickly. And then within the MRO manual or guidance manual, we've specifically addressed what is a valid prescription, when should it be used, is it used in the time period it was supposed to be used. For instance, you get a prescription for back pain 2 years ago, but now you're using it 2 years later, and it's because you hurt your foot. Was that prescription prescribed for the foot injury? No. So it takes into account many things as to what an individual as to how they're using a prescription. So a medical review officer, there's a lot of information they need to look at when it comes to prescription opioids. I think probably a good thing to point out here, and I was talking about this earlier, is one of the things that employers need to be very aware of is in applicant testing now, if your results are being reviewed by a medical review officer, you need to be very aware that a positive semi-synthetic opioid, that person should not necessarily be excluded from the applicant pool based on that positive, because that individual should or may have a valid prescription, using it according to the physician that's prescribing it, and for that. It does not mean that it's not necessarily a safety-sensitive issue in the position the person is applying for, but again, it was, I think, a little easier in a sense if it's a cocaine positive, that person was probably excluded from the application pool. If it's an oxycodone positive, that's a little bit different, and it should be reviewed by a medical professional to make sure it's not only a valid prescription, but the person is using it according to what they should be using it. So that was just one of the things that I wanted to point out. So again, with the question around that, it really is pretty detailed in the medical review officer manual as to what an MRO is to look at regarding what is considered a valid prescription. Okay, so I want to now launch into some of the studies that we've done. There's been several papers. I just wanted to point out some of them here that we have. And again, one of this is gearing up for the fact we wanted to give information, we wanted to gather information about the opioids, but we also wanted to look at not only what was a good concentration we should use as a cutoff around opioids, but why we use that concentration. So that's what I hope to explain as I go through here with a lot of these studies. Again, I don't want to delve too deep into the studies. What I want to do is a superficial look at it, and just so people explain, or just so I can explain why we set the cutoffs that we did. So the study overview, really we looked at oral fluid, blood, and urine. We collect all of these samples. These studies were IRB approved. They were done at Johns Hopkins University in the Behavioral Health Coordinating, or the Behavioral Health Center. And again, these were really good studies. We did several studies specifically around the opioids. The study goals are we looked at oxycodone and hydrocodone. We looked at kinetic relationships of whole blood and oral fluid and established or tried to establish concentrations in both. Did they correlate between what you see in the oral fluid and what you see in the blood, which primarily blood is used for impairing levels. So again, we try to look at if the blood has a concentration of this and the person is impaired, with the oral fluid also signified that the person was impaired. Again, with urine it's a little bit different because urine really is just the presence of the drug. They took the drug at some point, but it's very difficult to determine exactly when. Much easier within blood or oral fluid to look at the concentrations. We also looked at disposition of oxycodone and hydrocodone. We also wrote two profiling papers on oxycodone metabolites in urine. That's some of the information I'll share. And then the profiling of hydrocodone and the metabolites in urine, which really was the goal of some of these studies as we rolled out the mandatory guidelines in adding the semi-synthetic opioids. Dosing study, we used a single dose of Oxycontin tablets. They were 20 milligram oxycodone tablets. And specifically for hydrocodone, we either used 2 Norco tablets, each containing 10 milligrams, or 1 hydrocodone tablet containing 20 milligrams. And the milligrams in themselves are pretty important because that's really the top concentration that most individuals would receive for any pain management. That's a pretty significant amount. Most individuals will receive either a 5 or 10 milligram tablet. So we dosed at a pretty high level to get these concentrations. We did have 12 subjects. They were healthy adult volunteers. They were drug-free. And again, we had timed oral fluid and blood collections. And we pooled the urine specimens within the time, again, as presence of drug, and looked at that. And the individuals had 2 nights of resident stay, so we controlled the individual and what they were taking. The time course is just set out how we looked at this. We collected, again, oral fluid, blood, and urine on the left upper corner. You can see at the time intervals, basically it's 1 hour. It's right at the time that the person was given the dose or 15 minutes after, and then et cetera, all the way through day 3, which we looked at 50. We stopped the collection at 52 hours. Again, just the blood and the oral fluid and the urine, we collected all of those. I put this in here because specifically a lot of interpretation around the synthetic opioids, or the interpretation in general, is very important. Oxycodone, for instance, has 2 separate metabolic pathways that it can go through. They can go through one metabolic pathway called CYP3A, and they can go through another metabolic pathway called CYP2D. And this really is based on the individual and the individual's enzymes they have in the body. So it's very specific. So again, 2 of the major pathways, as I said, the CYP2A and the CYP3A, or CYP2D and the CYP3A. CYP3A would go to noroxycodone, whereas the CYP2D would go to oxymorphone. Very important. So if you have an individual who's telling an MRO, yeah, I have a prescription for oxycodone, and they see oxymorphone on the test results that come back from the laboratory, is that possible? Yes, very possible. Is noroxycodone possible? Yes, that's also possible. Also, if they see noroxycodone and noroxymorphone, which is another metabolic pathway that goes from noroxycodone, as the bold arrow indicates, it can also go to noroxymorphone. You could see essentially all 3 metabolites within an individual's urine in this case, and it would be acceptable that they would be on an oxycodone prescription. So very important. It's not as complicated when you're looking at codeine or morphine. It gets much more complicated when you're looking at the semi-synthetic opioids, specifically oxycodone and hydrocodone. So with that also, just in the review of the hydrocodone metabolism, the same thing, same pathways as far as the 2 enzymatic pathways, but you're looking at hydrocodone, which will go to norhydrocodone and hydromorphone. Again, if I can back up here for a second, so you're looking at hydromorphone here, I should use the arrow. So if you're looking at hydromorphone here from the hydrocodone, then when you back up, you're looking at oxymorphone here, but noroxymorphone. So there's distinguishing characteristics between those. But again, in all of the pathways between oxycodone and hydromorphone, you do have some analytes that cross over. So medical review officers really do need to look at that information and critically look at this information to make sure the person that's telling them, I'm on this prescription drug, that truly is the prescription drug they're taking and not another prescription drug that they're actually abusing. There was, within our studies, there were no serious adverse effects, but there was some mild to moderate adverse effects, including nausea, vomiting, lightheadedness, etc. And I mention this because, again, as I stated, is we wanted to look at concentrations would be about the top concentration that an individual or a naive user of these drugs would look at. And that also looks at the impairing part that I'll talk just about a little bit later. So again, there was some mild to moderate effects taking this dose. Now I'd like to look at the blood and oral fluid results. And these really are some of the correlation results of what we wanted to see within an individual, and then I'll go to the urine results. But again, as I talked about briefly before, when you look at quantitative levels of blood compared to oral fluid, you want to look at, we also, within these studies, we also looked at impairing factors for these individuals taking this dose. So we did what they call cognitive testing. There's a number of cognitive tests. It's performance testing around what they have as DSST tests, etc. There's a number of different tests, but the bottom line is you can show if your performance is actually hindered or enhanced by these drugs. So what we wanted to look at with looking at these levels of an individual, specifically here, when you're looking at oxycodone levels in oral fluid as compared to the blood levels here, was that person in this time frame semi-impaired based upon these tests, and what quantitative levels did you see in both blood and urine? So that's primarily some of the information we wanted to gain out of this study, is to show, is there any correlation between oral fluid and blood or impairing levels of what we see? And again, I should mention here that the federal program is a deterrent program. Nuclear Regulatory Commission is a fitness for duty, which means as a drug test, you're looking at it, is the person fit for duty? In the federal program, it's not looked at under fitness for duty. In the Department of Transportation program, it's sort of a little bit of both. It's not only a semi-fitness for duty, but it's also a deterrent program, but they also have a clinical component or a medical evaluation with that program. And that medical evaluation could include or looking at what prescription drugs that person is on. So again, I think to gain information on all of this when it looks at comparison is we wanted to understand, is there correlations here? This again was just the noroxycodone and the norhydrocodone levels, and again, as a very simple thing, these were basically the metabolites of the oxycodone and hydrocodone. So what we did is we correlated the metabolites of both of those in comparison between oral fluid and blood. And again, you can see that there's a much better correlation with when you look at oral fluid and blood levels in the actual concentrations here, which we're looking at nanograms in both of these samples. So again, as far as the metabolites, there's a very good correlation here. So this really is just an oral fluid-blood ratio and its correlations. It's specifically looking at the ratios. It's not really technical information. I think the thing to gain from here is when you look at oral fluid for oxycodone and hydrocodone, you can look at the oxycodone, the noroxycodone, oxymorphone, and noroxymorphone. So we looked at all of the the metabolites as well as the parent drug when it came to correlation between the two. In a summary of the blood and oral fluid results, again, looking at oxycodone and hydrocodone appeared in oral fluid and blood within 15 to 30 minutes after an oral administration. So again, as a practical point, if a person took a prescription opioid at work and it was under a reasonable suspicion test or a post-accident, yes, within 15 to 30 minutes, you would see these levels specifically in oral fluid. Second bullet point, because oxycodone was an extended release preparation, entry into the oral fluid was a little slower. A lot of these are essentially extended release preparations, so you would imagine it would be released slower in the concentrations, but the concentrations remained elevated longer, and that was an interesting fact with that. The profile and appearance and disappearance was similar, but the analytes were detected for longer times in oral fluid, unlike blood. Again, oral fluid concentrations were considerably higher for parent drug, which was from the first slide I showed, and in oral fluid compared to urine. And then the normetabolite concentrations were similar in oral fluid and blood. They both represented both of them relatively well with concentrations of the metabolites. And then, again, I think most of the rest of the information here is just for reading. But, again, part of a pilot study looking at blood hydrolysis is it did reveal somewhat higher concentrations of the oxymorphone, and that data wasn't shown. That's in a part is because within a person's body, you look at free drug as compared to drug that's actually been metabolized. So there is a difference in what you look at, or active drug as compared to drug that's been metabolized that's not active, and that really is around the hydrolysis part. So this is some of the urine results that I wanted to show. Again, I wanted to show this because there's a huge variability when you're looking at what we signify here as total oxycodone, total oxymorphone, total noroxycodone, and total noroxymorphone. So again, with these three levels here, the premise of this slide is you wanted to see if an individual in a urine sample, what the concentration with this, what the relatively highest concentration you would see with a single dose. And this really is to give medical review officers the information to say, if you had an individual that said, I have a total noroxycodone level of 2,500, which would be in about this area, is that expected from a single dose? Generally, yes. If you had an individual, say, that had a total noroxycodone level of 5,000, is that expected from a quantitative level? Again, 5,000 being up in here, you wouldn't expect that from an individual that was on a prescription medication that was taking it as prescribed. So again, we wanted to look at levels or concentrations of levels that we could see as a maximum level, and what the other concentrations of these other drugs fell under the curves of essentially what you would see as a quantitative level. Again, I know that's a technical part, but again, if you're looking at a prescription opioid and is a person taking it as prescribed, I think this is some additional information that medical review officers may want to look at that's coming from the laboratories. And again, just to point out here, this was essentially hydrocodone or the parent drugs as compared to the metabolites, and this is total. So again, relatively the same relatively the same concentrations here, just as to point them out, they look relatively the same. A little lower on the hydromorphone levels than when it comes to the oxymorphone levels here. This slide, all I meant to show here is there is a huge variability between donors. These were all the donors that we used. And again, as you can see, this one individual here was very, very large when it comes to hydrocodone excretion. Why? Again, were they excreting it through a different metabolic process, either CYP2D or CYP3A? And again, but that's something that an individual has to take into account, or a medical review officer has to take into account, is what concentrations could you see? Again, this was pointed out when I was giving a presentation for occupational physicians, is again, there's a number of other drugs that also could interact with synthetic opioids, which you need to be aware of. So if you start to take, for instance, a new blood pressure medication, could that have influence on the semi-synthetic opioids that you're taking? And for this reason, the physicians were asking is because in taking a new drug, could that actually cause impairment of a drug that you've been on, which it very well could if it increased the concentration. This again, as I pointed out, the simple part of this is we wanted to look at cutoff concentrations of where we should establish them. And as a federal program, a lot of the private entities use the federal standards to establish the cutoffs. And again, as laboratories move to standard cutoffs around what we've established because of cost and efficiency and running samples all the same way, we wanted to establish a cutoff concentration for laboratories of where not only could we see, this is detection time in hours, you could see up to 30 hours. So a detection window, and again, this is on a single dose, for instance with oxycodone. So we wanted to establish a relatively long detection window with a concentration that could be easily looked at across the board for oxycodone, oxymorphone, noroxycodone, and noroxymorphone, the 4 analytes that we looked at. So again, established from here, we looked at a cutoff concentration between 300 and 150, and we chose ultimately the 300 concentration. There isn't much difference. And within this reproducibility at a 300 cutoff, you had a great correlation of what was detected as a positive screen was also confirmed. Some individuals looked at the concentrations that it was relatively low, but what we wanted to do is be able to give the medical review officer the information, and then they can determine was that an opioid that the person should be using, a valid prescription, not necessarily set a cutoff at 2,000 where you're ruling out so many individuals that you wouldn't even see a person after 3 hours if they were taking a single dose. Again, within that 3-hour period or longer, could that person possibly still be impaired or on the job, and should they be tested? So again, that's why we chose some of the concentration levels or the cutoff levels that we looked at and why we looked at those. Again, this was hydrocodone. We looked at, again, we wanted to have a relatively long window of detection, around the same for both analytes. So again, what we chose was 100 as a cutoff concentration, which again, with at least the norhydrocodone, you at least get out to 33 hours of time that you could detect that in a single dose. And again, I did want to mention that within all of these, when we were looking at cutoff concentrations, you can look at a concentration, but it's very hard to determine specifically in urine that this was from a single dose, when the individual took it. Most prescription medications aren't given as a single dose. The person is on a regimen of they take 1 oxycodone at night, or they take 1 during the morning. Again, it wouldn't necessarily mean that you could exactly look at when it was actually taken specifically in urine, because again, urine points out that it's present, not necessarily what the actual level was in the blood or oral fluid when it looks at was it a safety-sensitive issue? And again, the other part of that is, as you're taking multiple doses, these concentrations remain high over the length of time you take the prescription medication. This was just establishing. We looked at, again, just to evaluate the evaluation of the analyte by the cutoff is when you actually look at the number of samples, this being the number of samples, 120 samples being taken for the specific drug, is when you set up cutoff concentration, how many of those samples do you actually sweep in? Again, when we looked at 100 specifically for, I think this is oxycodone, pretty much you gain at least the majority, probably 80, 90% of all samples will test positive at that cutoff. Again, if you set the cutoff here at 2,000, you would barely get 20 samples out of the 120, or a little over 10% of those samples you would detect. So again, in a program where you want to give the most information to the medical review officer, then you want to look at something that establishes a cutoff that's going to give you the amount of samples you want to see as a positive. Again, this over here just looks at the hydrocodone, again, based on concentrations and cutoff concentrations with the analytes that we set. So the results overview for oxycodone and hydrocodone, really, essentially, I think I pointed most of these out within oxycodone and also hydrocodone. There wasn't anything specific that came out of it except I think one of the things, all blood specimens immediately before oxycodone dosing were negative and less than LOQ for analytes. And then oxycodone was detected in specimens from the first collection period, which was 0 to 2 hours, for 11 of the 12 subjects. So again, I think it's a pretty reasonable marker. You're going to get, if an individual at least takes one tablet, you are going to get a positive at least from that person under the program currently. And then hydrocodone and norhydrocodone were initially detected in the majority of subject specimens after 2 hours of drug administration. And then hydrocodone was the most frequently detected in combination with norhydrocodone. So again, since we look at all four analytes, it's pretty reasonable that we would detect at least the largest frequency of semi-synthetic opioids that are not only taken but also abused in the United States. In summary, again, I won't go over this, but again, just to point out the fourth bullet point, overall, this data suggests that monitoring of oxycodone and oxymorphone would be effective at cutoffs up to 300, so that's why we chose the 300 level. And again, under hydrocodone, we looked at a screening level of 100, where we detected most of the positives. And again, under summary of hydrocodone, overall, this data suggests that drug testing requirements for hydrocodone should include hydrocodone and hydromorphone in hydrolyzed urine. Reason behind that is the peak concentration of the metabolite is longer as a window of detection. Also, the concentration is larger. And again, so we've looked at that, and that is part of the mandatory guidelines to look at those. It does give more information to the medical review officer to make sure that the person is on the prescription or the valid prescription they say they're on. Okay, so real quickly, just to touch on MRO manual, the guidance manual that we have, we do address the addition of the prescription opioid drugs in the drug testing panel, the specific 4 that I showed. And then the biggest question I think we looked at a little bit earlier is what is considered a valid prescription under the Drug-Free Workplace Program, and how will it be interpreted by the medical review officer. So again, I don't want to go into the specifics, but some of the information that I pointed out from the MRO guidance manual here is, and again, it's how a prescription, from the bottom here, it's how a prescription can be given to the medical review officer, how the medical review officer can talk to the prescribing physician. Also, if the individual MRO can verify that prescription with the pharmacy. Again, there's a number of different things we looked at to be able to have the MRO find out the information from the donor. And again, I think the premise of it starts here. If the donor claims to have taken a prescribed medicine that contains either the drug reported positive or a substance that can metabolize to the drug, the donor should provide the medicine container with the appropriate label prescription on the label with the container with that person's name. Again, there's many questions around if an individual is taking it under, is it a valid prescription? Is it the time intended? Was the prescription is taken for the intended use of that prescription, etc. So there's a number of things that the medical review officer will have to look at when it comes to that. The second part of that is something I was mentioning here, and that's at the bottom here in red, is when determining whether a legitimate medical explanation exists for a positive test, the MRO may consider whether a medicine was used during the time period for which it was legitimately prescribed, if the donor was not, or if the donor's use was not medically authorized, the specimen would be reported as positive. Now, we've been in several discussions. If an individual, if it is his prescription, it may be 3 years old. Was it prescribed for that person? Yes. Could it be legitimate or being legitimately prescribed? If you take it 3 years later, it probably wasn't prescribed for what the person is taking for it now. But should it be reported as a positive? Again, or should it be a safety-sensitive issue? So I think with those, the MRO has to weigh, is it a safety-sensitive issue? Was it being taken appropriately? And then they have to weigh that. Does it have an impact on the job? And I think that's how employers should also have to look at it, because it does weigh on the fact that is it a safety-sensitive issue? And with that, some of the current main points is under current conversations. Again, part of the regulations are dealing with marijuana, synthetic marijuana, and the opioids. A question came in: if an employer uses the previous DOT CCF after June 30th, 2018, will this invalidate the test? No, it will not invalidate the test. What it actually says is, in DOT's guidance, is after June 30th, if it expired, and these are expired OMB approved forms after the 30th of June 2018, is a memorandum for the record will have to be obtained by the collector. The test will not be canceled. It will not be rejected. They just have to obtain a memorandum for the record. So again, there is an ever-changing landscape of marijuana use and regulations. It's a catch-22 on the creation versus detection of the synthetic marijuana types. Specifically, they seem to be moving targets. And then the prescription opioid-related deaths are staggering, and that's one of the reasons we're trying to get a handle on this, essentially in the federal programs also, is because we know there is a prescription or a semi-synthetic opioid crisis in this. Major changes, as pointed out early, is I think it's important, specifically as oral fluid comes out. It's going to be an alternate matrix, but I think it's a very beneficial alternate matrix, and it will be under the federal program. So it's basically got the federal standards that most of the laboratories will move in that direction as they look at targeting the concentration for the cutoffs for the screens and the confirmations, as they did in 2004 when we put out the proposed. Again, I think pursuing different matrices for different purposes is very reasonable, and I think, as I mentioned before, an effective program has, you have to have alternate specimens so that basically people can't beat the drug test. And again, through a lot of the studies we have done, not only around opioids but marijuana, is science really should lead the policy. Sometimes that doesn't always happen, but again, it should. And I think the extension of the oral fluid mandatory guidelines as far as federal standards also have very promising use within driving under the influence of drugs or roadside testing, etc., using oral fluid in the devices they can use there. So I think there's a lot of reasons behind oral fluid testing. With that, I want to thank you, and I know I've went over a little time. Again, at the end of this presentation, I'll just go to the next slide real quick. I've detailed some of the actual revisions to the mandatory guidelines. You are free to go through those. Look at those. If there's any questions you want to pose later, I'm sure we'll be able to gather those. But these are a little more detail of what we actually changed along those type of mandatory guideline changes that we made. So with that, I'm going to turn it back over to Julian for questions, if we have questions, and Natalie, and we'll be glad to answer any questions you have.
Great. Well, thank you all. Well, I think that since we are out of time at the moment, we'll just end it there for now. If anybody has any further questions for NSC as we continue to provide support to our members on this and other issues, please feel free to email me, Julian, Julian, .Hoffman, Hoffman, @nsc.org. I know we'll be putting this webinar up on the site for future access, and we hope that it'll prove useful to you as you design your own routes forward for your companies. We look forward to seeing you on another webinar soon. Everybody have a great rest of your week.
Thank you all for joining us for the webinar, and we appreciate your attention and participation in today's event. You may now disconnect.